welcoming Caitlin Falconer to the stage. So Caitlin's at UPCC. Yes, great, excellent, and has kindly agreed to take on a talk called Genomics 101. We got a lot of feedback. This is the second time we've run this workshop, and we've got a lot of feedback saying that the pitch was too high last time and need to bring the level down. I certainly struggled to understand a lot of the content that I myself was presenting, so that's all. But that may not be related to the content, it might be related to me. But Caitlin's kindly summarising genomics in 20 minutes.
Hopefully I haven't made it too long.
Fantastic. Excellent. That's great. Thanks for coming on, Caitlin.
Thanks. Thank you. So I'll be giving a bit of an introduction into genomics, but I'll be giving it from a bioinformatics perspective. So I'll go through two types of sequencing platforms that are available and when you might decide to use either of those. I'll talk about the difference between using short and long read sequence data. I'll talk about the role of metagenomics in place of culture-based methods. I'll go through a typical bioinformatics analysis pipeline. And I'll give an introduction into phylogenomics and clustering analysis. So I've got a scenario here. We've got a collection of multi-resistant organisms, and these have been taken from hospital inpatient screening swabs, from some clinical specimens, and there's also some environmental samples. So some of the things we might want to know about these samples are, what are they? What's the species? What's the strain? We might want to know what types of resistance mechanisms they have and how related are these organisms. So could this collection be part of an emerging outbreak? So we can determine what type of organism they are and what species they are by using some traditional PCR-based multi-locus sequence typing methods, but this really only gives you a small window into the genetics of an organism, and at this level, things can look pretty similar. By whole genome sequencing, we can see the full picture into the genetics of an organism. So this gives us a much higher power to be able to discriminate between these strains. And using whole genome sequencing allows you to perform other analysis for gene content like resistance determinants, plasmid types, virulence factors. So when you whole genome sequence a sample, the sequencer generates some files containing short strings of DNA sequences called reads. And using these reads, we can map them to a reference sequence and look for differences, such as single nucleotide polymorphisms. And when we're looking at how related strains are, what we're looking at is whether they share some of those same SNPs. From these reads, we can also attempt to reconstruct the original genome sequence, so we can try and string them all together based on the overlapping regions of similarity and try and reconstruct that original sequence. So there's a number of different sequencing platforms available. Two of these are Illumina and Oxford Nanopore. So Illumina is the most widely used sequencing platform at the moment. There's a couple of different machines within the Illumina family, but generally they're all quite high throughput. The data that you get from them is high accuracy and they're low cost to run. The downside of Illumina is that the reads are quite short, in the range of 50 to 300 base pairs. So to put that into perspective, an E. coli genome is about 4.5 million base pairs in length, and the human genome has 3 billion base pairs. So Nanopore is a newer technology. It's been around for less than 10 years, and it can produce reads that are orders of magnitude longer than Illumina. And you can also perform the analysis in real time, meaning that as soon as the sequencer starts, you can start analysing the data as it is produced rather than waiting for the whole run to finish. Nanopore data, however, is of lower accuracy, and it is a fair bit more expensive per isolate to run at this stage. So why is read length so important? Well, in a genome, there's lots of repetitive and duplicated regions, and there's also mobile genetic elements like plasmids. So with only small reads, it's quite hard to resolve those repetitive regions. It's hard to know where they occur in the genome, how many times they repeat. And the circular nature of bacterial genomes also adds complication when you're trying to assemble the sequences together. So it's like having a jigsaw puzzle with lots of tiny pieces, lots of these repetitive and duplicated regions, having no edges or corners, or even knowing precisely how many pieces are in your puzzle. If we have long reads, however, that span across those repetitive regions, it's much easier to assemble the full sequence together and resolve the full genome. And in the case of bacterial genomes, if you've got longer reads, it's easier to resolve the plasmid, so you can determine which genes are from a plasmid and which are from a chromosome. So if long reads are so good, why don't we just use them all the time? Well, as I mentioned, nanopore is a bit more expensive, and the accuracy of the nanopore data can have quite a big impact in the analysis. So when you're looking at those very precise mutations in the genome, or if you've got a handful of, or if your genomes only have a handful of differences between them, the accuracy of the long reads, the lower accuracy of the long reads can have quite a big impact in the results. So what we can do is we can use both the high accuracy Illumina data with the long reads and try and assemble high quality complete genomes by using a hybrid of the two. So traditionally, infectious disease surveillance and diagnostics is done with culture-based methods. So an alternative is using metagenomics, and this is where we can extract the DNA directly from a sample and sequence that DNA. So it allows you to capture things that are in the sample that might have been missed or might have dropped out during a culturing step. Metagenomics can also speed up the analysis, so you're not waiting for that culturing step. You can process the sample straight away. So long reads is also useful for metagenomics. So for example, if you take every fish in this picture as a different type of organism, with longer reads it's easy to determine where the read came from, which organism it came from. And so if you're looking for a particular resistance gene or you've detected a resistance gene, using longer reads it's easier to determine which organism that came from and whether it was from a plasmid or from a chromosome. So there's a couple of great examples of whole genome sequencing being used in infectious disease surveillance and diagnostics. So whole genome sequencing was used to confirm ceftriaxone treatment failure as opposed to reinfection in a gonorrhea case. And this study highlighted the utility of the higher, the utility of having more data to be able to determine this treatment failure as opposed to the traditional multi-locus and multi-antigen sequence typing methods. Nanopore was recently used to confirm 2 new cases of CPE that occurred in a regional hospital, and these were then linked to some previous out cases in another hospital and provided actionable results to the infection control team. And nanopore metagenomics was also used recently to diagnose a slow-growing organism from a bacteremia that resulted from a dog bite and may have rapidly sped up the diagnostic time from 6 days down to 19 hours. So back to our scenario, we've covered that we can use whole genome sequencing to identify what strains we have. We've decided that Illumina sequencing is good for high accuracy data. If there's any important or interesting isolates, we might nanopore sequence them so that we can assemble some high quality complete genomes. We might also use nanopore for the real-time results if we have any urgent samples that we want to know about. And we might also use nanopore for metagenomic analysis of that clinical sample or of any environmental samples. And so how is all of this data analysed? How do we determine what strain they are? How do we determine what the resistance genes are in there? So this is a typical bioinformatics pipeline. This one's specifically designed for culture-based Illumina data. And so once a sample is sequenced, it produces a number of files containing those sequence reads, and these files are called FASTQ files. So they contain the string of the sequence, and then they also give a bit of quality. So the first step in any sequence, in any pipeline, is quality control. So we look at how good those sequences are. And this involves cleaning off any bases that have a lower quality score. So we're not really confident that what the sequencer detected is actually what's in there. So what we do is we trim those bases off. So this is particularly at the ends of the reads around here. If we're not confident that that is what the sequencer says, we trim them off. So once we have our set of high quality FASTQ files, we taxonomically profile them. So this involves searching for matching sequences between the reads and a large database of reference genomes and determining the most likely strain ID. The rest of the steps in the pipeline are all performed according to what strain has been detected, what strain has been assigned to that genome. So this includes SNP profiling, where we align those reads to a reference genome and we look for those single nucleotide polymorphisms, so those SNPs. We can also determine what MLST they are. We look at antibiotic resistance determinants, and we can also attempt to assemble those reads together into an assembly, and then we can perform further analyses for more antibiotic resistance. We can confirm our MLST profile if we wish. We can look at plasmid types and virulence factors on those assemblies. All of those processes in the pipeline are all performed automatically. So we feed the read data in, the read files in, and then all of the rest of the steps are done automatically. So once we've got that, from our SNP profiling, we can do phylogenomic analysis and cluster detection. So this is where we're looking for how related those strains are. And then we can combine all of that information with data, with patient clinical data out of the REDCap database to produce a report for the ID and infection control teams. Now, a caveat to all of this analysis is that because the taxonomic profiling, resistance detection, MLST profiling, plasmid typing, all of that is done based on existing databases. So the results are dependent on what database you use, how well that database is curated, how comprehensive the database is, and it can also be dependent on the version of the database. So as new things get added to the database, if you're comparing results from one run to another and the version of the database has changed, it's important to keep that in mind. So this is what a report might look like. This one is specifically for ESBLs and carbapenemase producing Enterobacteriaceae. So it gives us some of that patient information. It tells us what organism they are, the ST type, and also gives our gene profile. So this is just the beta-lactamases. And we have a bit of a summary about some of the important highlights that came out from this sequencing run. So here we've got some evidence of patient direct transfer between two patients of a Citrobacter, which have been highlighted in the report. And there was also ongoing evidence of transmission of E. coli ST131 within the hospital. So if we look at our phylogenetic tree, this highlighted clade here are samples that are all highly similar to each other, and they occur just within the hospital. And we've got 2 new isolates that have occurred within this clade. So this suggests that there's some ongoing transmission of this ST131 type. So a phylogenomic tree is a representation of evolutionary relatedness of samples, and it uses single nucleotide polymorphism, so those SNPs, to perform a pairwise comparison between all the isolates. And isolates that have some shared SNPs will occur on the same branch, and the shorter the branches between 2 samples, the more closely related they are, so the fewer SNP differences they have. So if we look at our 2 highlighted groups, these ones here and these ones here represent some highly related isolates, and this group here in particular showed no SNP differences between them based on this analysis. Branch order in a tree doesn't matter, so you can freely move these around into any order, or you can switch entire groups of branches. So this tree means the same as this one. The shape of the tree also doesn't matter. So these 3 trees all mean the same thing in terms of the relationship of samples A to D, E. It just depends on if you're telling a different type of story, you might use a different shape of tree. So if we're trying to highlight that there's a specific clade, we might use the rectangular tree. Or if we're talking about a transmission network, we might use a tree shaped like this. So when we're calling SNPs and performing a phylogenomic analysis, the selection of the reference is really important. So if we have a closer reference that's more similar to the samples, we have a larger core genome, and the core genome is the area that is shared by all samples. So this is all the samples have all of these genes present. So when we're performing the analysis on a larger core genome, we've got more information, and we can see more of those differences between samples. If we use a more distant reference, the size of the core genome can become smaller. And so we're comparing less of the overall genome, and samples might appear falsely similar because you're only comparing a smaller proportion of them. So if we come back to our tree from before, all these samples looked identical based on SNPs in the core genome, but are they really identical? So we could look at differences in the accessory genome. And because a phylogenetic tree is built using just SNPs, there might be other types of variants present. So there might be some small insertions or deletions known as indels. There might be large gene deletions, or there could be some chromosomal rearrangements. Now, the reason we only use SNPs in a phylogenomic analysis is because SNPs accumulate in a bacterial genome in a fairly clockwise and predictable manner. So we can be reasonably confident that a sample with 2 SNP differences has a more recent common ancestor than 2 samples with 5 SNP differences between them. So cluster analysis is an alternative method of looking at the relatedness between samples. So this CAFI has been developed to visualise clusters of samples depending on a user-defined SNP threshold. So we can give the maximum number of SNP differences that are between samples, and it will draw links between them in this cluster graph. And this is useful for visualising an outbreak. And because this is also using SNP differences, it's dependent on the reference. So if we did use a reference that was more distance, and we're comparing less of the overall genome, we might miss some of those SNPs, and 2 samples might have a link falsely drawn between them. But if we reanalyse the data with a more appropriate reference, we might see more of those differences, and that link might disappear. So overall, we've looked at species and strain identification using whole genome sequencing. We've determined the presence of resistant determinants and virulence and plasmid types. We've looked at SNP-based clustering and phylogenomics, and this is useful for identifying any potential outbreaks. And all of this genomic data can be combined with the patient metadata, and that can help us infer when, where, or how potential transmission may have occurred. Thank you.